All right, so this chapter, chapter 16, is going to be all about DNA structure and function. And you can't really talk about DNA structure and function without starting to talk about RNA as well. So this is a little bit of a throwback to chapter 5, where you started learning about macromolecules. And these are, of course, our nucleic acids that we learned about back then, but now we're going to go into much more detail. So DNA, of course, stands for deoxyribonucleic acid. Unfortunately, you do need to be able to recognize that term. Whereas RNA stands for ribonucleic acid. They're called those different things. So deoxyribo refers to the sugar that makes up the nucleotides within DNA, whereas the nucleotides within RNA have a ribose sugar. So you don't need to really worry too much about that. Just know that they're called those things for different reasons. It's not just all arbitrary. So these are the two types of nucleic acid that we've learned from chapter 5. And they're what we called polymers. So if you remember from chapter 5, all of the macromolecules to some degree had chains of individual monomers, right? So like your proteins were actually polypeptides that were made up of all of those amino acids strung together with peptide bonds. Okay, so this is the same thing, except now our monomer is a nucleotide. And if you chain together a bunch of nucleotides, like I've done in this image here, so here's a nucleotide, here's a nucleotide, here's one, and here's one. That is a chain of nucleotides called a polymer. And when they're nucleotides and you chain them together, we make a nucleic acid. Okay, so that's one of the four major biomolecules. So I'm just zooming in here on one of those monomers. So I'm taking, like, let's say T, for instance, right here. I'm taking that and I'm zooming in. And so this is like the biochemical form of what you'll see for a nucleotide. You'll see there's a sugar, you'll see there's a phosphate group, and you'll see that there's a nitrogenous base. And so in this case, this is the nitrogenous base for adenine, so A. So this guy right here. Okay. This is what he will look like chemically. All of these straight lines are, of course, bonds. You guys remember all that stuff. But all nucleotides have a 5-ringed sugar. Okay, so that's why it kind of makes that pentose shape, that pentagon shape. And then there's a phosphate group. We usually represent that with a circular shape, and like often in this simplified diagram right here. Okay, so here I have my phosphate group. Sometimes you'll see that there's a P in there. Sometimes you'll also see a minus sign because there's that O negative right here. And then there's a nitrogenous base. And usually the nitrogenous base is represented on the side like this. So this, in this case, because it's adenine, would be an A. Okay, so these are like your standard shapes. This is pretty much what you'll always see, this simplified diagram of a nucleotide down here. I do not expect you to be able to draw this top diagram. It's too complicated, and it's not worth your time for AP Bio. So there are going to be 5 nitrogenous bases across both DNA and RNA. Okay, so right here, these top 4, so C, G, T, and A, are going to be found in DNA. Okay, so these are your big 4 for DNA, and obviously DNA is pretty dang important. Uracil is going to be unique to RNA. So this guy right here, uracil, that is only found inside of RNA. And that's mainly because of just the chemicals that, the chemical bonds that make up the RNA, like that, excuse me, ribose sugar difference. There are no thymine nitrogenous bases in RNA. So there's mostly overlap. So these 3 that I'm underlining right now, C, G, and A, those 3 are common between DNA and RNA. But thymine is unique to DNA, and uracil, U, is unique to RNA. So just a little bit of a recap from chapter 5. When you start to string together nucleotides to form nucleic acids, you're going to be removing some water. Remember that, like, when we string together monomers, we have to remove a water. We remove an H from one area and an OH from another. In this case, you're removing the OH from the phosphate group of one nucleotide and the H from near the sugar on the other nucleotide. And that's going to create this chain. So we're going to be removing water. Remember, when we remove water, anytime you think about removing water, you should kind of picture like condensation around like your glass of water or something that you have out, right, on a hot day. So, excuse me, if you're removing water, we call it a condensation reaction. because water is going to be forming on the outside, just like it does when water condenses around your glass. Okay, so this is all review. With nucleic acids, the bonds that connect them are called phosphodiester bonds. So remember with proteins, for instance, we had peptide bonds. This is going to be phosphodiester bonds. This is just what we call it when you connect two nucleotides together, whether it's in DNA or RNA. So this is the classic double helix shape that you've seen a million times, I'm sure. So this shape right here is the shape that DNA is found in. And it's often referred to as a twisted ladder in addition to the double helix term because it looks like a ladder that's been, you guessed it, twisted. And if I were to stretch those out and make them nice and clean, this would be a nice simplified diagram of the nucleotides that make up those two strands of DNA. Okay. So the phosphate groups, the circles, and the sugars, the pentagons, make up the backbone. So those are like on the outside of the DNA. So that's like what's going on right here. Okay. Whereas the nitrogenous bases are on the inside. Okay, so all the C's and G's and A's and T's, those are all on the inside of the DNA. It's almost like it's protected, okay, because that's where all that information is, and we'll get to that in a minute. But just remember that you have a sugar phosphate backbone, so sugar and phosphate, right? That's why it's called that, because it's made up of, you guessed it, sugar and phosphates. And one thing you may notice about this diagram on the left here is that the pentagons kind of act like an arrow. Okay? And these arrows are all pointing up on the left side, whereas on the right side, they're pointing down. And that's going to become really, really important. DNA is made up of two strands, right? The left and the right strands. These bonds right here are actually quite weak bonds that are connecting the nitrogenous bases. Those are not very strong. They're not covalent bonds. Okay, they're just simple hydrogen bonds. And so we're going to later see what happens when you separate the DNA strand. But for now, I just want you to focus on the fact that we have two different directions of these strands, and that is called anti-parallel, because they are parallel. They're never going to touch each other if you extended those arrows all the way, but they're going in opposite directions, so that's why we call it anti-parallel. Now, your book is going to talk about the differences between the C to G and A to T base pairs. So, for instance, C to G has 3 hydrogen bonds that are going to form between C and G. And A and T are going to have only 2 hydrogen bonds. So like imagine this is A and this is T for now, just to simplify things. Okay. So there's only 2 hydrogen bonds here. So technically the C to G is a stronger bond. I don't need you to worry about that. Okay. They are not, these two strands are not bound together covalently, as I mentioned before. And that's going to become really important. This is kind of like a zipper. So this is all held together kind of like a zipper. And so if I unzip all of these little bonds, I can separate the strands. And that's going to become important when we get to DNA replication in this lecture. So again, I mentioned that they are anti-parallel. I find it best to just picture the pentagons as kind of being an arrow. Okay, and so your arrow is pointing in one direction. And excuse me, you will see in the textbook that the book makes a really big deal about the 5' end versus the 3' end. Okay, because these are just naming the different carbons that are there. That's why it's called 5' and 3'. It's just naming the carbons that are there in the pentose sugar. And I don't want you to freak out too much about it. Just know that we typically picture the arrow head going from the 5' end to the 3' end. That is actually really important. The 5' and 3' directionality is actually really, really important when we get to DNA replication. And you're going to see why in just a few minutes here. But I don't need you to be able to recognize where the 5' and 3' is if I give you like an intense diagram like this one with no labels or anything. I mainly just want you to understand it conceptually. So a couple of differences between DNA and RNA. So DNA is this double helix on the right here, right? And DNA has 2 strands, right? So that's what makes it that ladder look. It has 2 strands, a left strand and a right strand, depending on, how you're looking at it. Whereas RNA is going to be single-stranded. It doesn't like to hang out with anyone else like that. Okay? So DNA is pretty dang stable like this, whereas RNA does not like to hang out with anyone for a long time. Okay. So I mentioned this earlier, but DNA has deoxyribose sugars, whereas RNA has ribose sugars. That's why they're called different things. Okay. And finally, just as a result of those sugar differences, DNA will have thymine as a nitrogenous base, one of its four, whereas RNA will have uracil as one of its four nitrogenous bases, whereas it will not have thymine. Okay. And so I've mentioned all those before, but here's a nice table. I would add this to your notes, this table of the differences between DNA and RNA. And these are all structural differences, right? Not functional differences. So in these bases, as I mentioned earlier with the A to T, C to G thing, we have all of these hydrogen bonds here that could be broken. Okay? And that occurs with RNA too. Okay, and that's going to become important when we get to DNA replication and gene expression, so chapter 17. But these pairings are ubiquitous. They're found in every living thing on Earth as far as we can tell, right? So in every organism, A binds with T in DNA and C binds with G in DNA. Okay. So your book will talk about the difference between purines and pyrimidines. It's really not that big of a deal. Basically, purines are a little bit bigger. Okay, so there's A, so adenine and guanine, A and G, and there's thymine and cytosine. And so these guys like to hang out. The A and T like to hang out, right? They have 2 hydrogen bonds between them. The G and C like to hang out. They have 3 hydrogen bonds between them. And so the best thing to do is to just picture the fact that if you had a bunch of these, they would all have the same width, because like every single time you have a big boy, so like here's my A, that's a big guy, and a small boy, so here's T, he's small, and so they're going to hang out. And so I have a big boy and a small boy, they're going to be next to each other, so that makes 2 plus 1 is 3, right? So I'm just going to say that this has a width of 3 because there's like 3 rings here, whereas G is a big boy and C is a small boy, so here's like 2 rings, here's 1 ring, and then that's also going to have a width of 3. So this is why DNA has a consistent width all the way around. It's not a ladder that has, it goes kind of wider and narrow and wider and narrow. It's going to be all the way down the same width. And you don't really need to be able to remember which ones are purines and which ones are pyrimidines. Just know that it's a thing is really all I expect you to be able to know. And it's just called this because of the different structural rings that you have inside of those chemical bonds. So like you'll see here, adenine has this ring and this ring, and so that's why we represent it with all of those different geometric shapes, whereas thymine only has this ring. Okay. So you'll notice again that we have that 5' to 3'. We always point the arrow from 5' to 3'. And again, I don't need you to be able to recognize which one's 5 and which one's 3 without it being labeled at this point. Just know that those 2 strands go in opposite directions. If it wasn't for that, it wouldn't work, and we'll explore some of that in class. But our RNA, okay, so now we're switching gears over to RNA. So we're not going to have thymine anymore. We're going to have uracil, a U, okay, has 4 major types that we care about. And really, some of these we care more about than others. So like mRNA is huge, very, very important. So that stands for messenger RNA. It's going to become really, really important. tRNA is going to be really important. Both of those are going to be really important when we get to chapter 17, as with rRNA. And so the R stands for ribosomal. So you remember from, I believe it's chapter 6, what a ribosome is. And that's going to become really, really important when we get to gene expression as well. Okay. So basically, the mRNA is a messenger RNA molecule, and a messenger carries information, right, from one place to another. In the case of DNA or gene expression, so that's like where we take our information. So we have our DNA in the nucleus, right? And I actually use the information in the DNA. What I'm basically going to do is take the information that's in the DNA and copy it onto some mRNA. So like the DNA is kind of like a Latin cookbook. Like that's great and all, but I can't read Latin. So I don't know how to make the recipe that is written in the DNA. But if you transcribe it into English, so that's what's happening here. My mRNA is kind of like my English cookbook. Okay. and I'm going to transcribe it, and then eventually I can finally make some food. I can finally make a protein with that information. This is my recipe now, right? I can make that at a ribosome. And so that's going to be the process of translation. I don't need you to know this like to the nth degree at this point. This is really going to come into its own during chapter 17, whereas we are in chapter 16 right now. But I'm just giving you a little bit of a primer on where we're going. Okay, so rRNA makes up your ribosomes. Okay, and so your ribosomes are actually what will make the proteins. And so rRNA it's a really intense RNA molecule that kind of folds in on itself and becomes really, really important during translation. But the RNAi molecules are going to be involved in regulation of gene expression, which sounds really, really scary, but we have an entire chapter for it, chapter 18. And so I don't need you to freak out about the regulation of gene expression yet. Basically, it's like, oh, sometimes I want proteins and sometimes I don't. Okay. Here's some information about structures of DNA and RNA that I think would be really useful, some supplemental videos. It's really important that you understand the structural differences between the two before we start getting into the functional differences. Now, to that end, we have unfortunately quite a crucial process that we need to learn in chapter 16, and that is the process of DNA replication. So DNA replication is pretty involved. There's lots of different moving parts. There's lots of different molecules that are going to be involved, and it kind of helps to always keep in mind why DNA matters. So DNA matters because it has our information, and that is the information that we can use to eventually make a protein. This right here, DNA, gets transcribed to make RNA, which then gets translated to make proteins. That's like my Latin cookbook getting transcribed to an English cookbook. So now my RNA is my English cookbook, right? And now I can actually make the food, so that make the protein. That is what's called the central dogma of biology. It's called that because this is like basically why every living thing works the way it does, because we have this information and we have a means of using that information. And so it's important to take a step back and realize why DNA is so important, because we're going to start replicating or copying it. And you guys have seen this before in mitosis and meiosis. And so we know that DNA is important, but now you know kind of a little bit of, as to why. And so when we copy DNA, that's a really big deal. We're going to make extra copies so that all of our cells will have it, and we can even reproduce cells, right? So DNA replication occurs during S phase of interphase, as you guys remember. And this is going to be really, really important to do before mitosis or meiosis, depending on where in the body you are, right? And so DNA replication has three major steps. So remember, DNA is in its nice little double helix shape. And in order to copy it, we really need to kind of unwind it. We need to open it up. Okay, so there's an unwinding stage. So this is kind of like unzipping. Remember I said it was like a zipper earlier. This is like unzipping the DNA. And there's going to be some enzymes that are important during that process. So there's helicase, there's topoisomerase, and there's RNA primase. And I don't expect you to memorize what those do at this point. We're going to be diagramming them on a foldable together, because that's kind of the best way to learn this, is to just practice and diagram it over and over and over again. Then we have the synthesis stage. Okay, and so this is where you actually synthesize or make your DNA molecule. So we're going to get to that, your DNA strand. Okay, and that is going to involve DNA polymerase. So polymerase, it ends in ase, so it's an enzyme, right? Just like all those things up there, the helicase, the topoisomerase, etc. But a polymerase is responsible for polymerizing or chaining together things. And so if I look at the name, it's probably going to be responsible for chaining together, you guessed it, DNA. Okay. And then our final stage is ligation, and that's going to be done by an enzyme called DNA ligase. So the nice thing is sometimes we get really lazy with our naming, and so we can just name the enzyme after exactly what it does. Okay. So during the unwinding stage, so I'm just zooming in on this first stage right here, unwinding. My DNA is all tightly coiled, and that's great. Like DNA should be in its coiled double helix form most of the time because that helps protect all of that really, really necessary information. Remember, all those nucleotides are on the inside. But if I want to copy the nucleotides, I want to copy that information, I kind of need to access it, right? You can't access what's inside unless you open the door. And so replication is going to start by unwinding the DNA. You're kind of opening it up. You're letting stuff access it, which is a little risky, but it's necessary if you're going to create a copy. So at the start of replication, your topoisomerase is going to begin unwinding the DNA helix. Topoisomerase is it's got quite a few jobs during this process. One of the biggest jobs that it has is that it kind of stabilizes upstream. So like right here, I have DNA helicase. DNA helicase is going to create what we call a replication fork. So it's basically going to do most of the unzipping. DNA helicase does most of the unzipping of those two strands. So that's what's happening here. I have one strand and one strand. They were together right here, just like we see upstream, we call it. But down here, we've started to unwind them. Okay. And basically, DNA helicase is just going to start breaking the bonds between those nucleotides. It's going to start saying, A and T, I don't want you guys hanging out anymore. Okay, so like if this was A and this was T, they really liked to hang out. They had those hydrogen bonds connecting them, but then DNA helicase is just like, no, I want you guys apart for now. Okay, and then topoisomerase is again kind of going to stabilize ahead because remember, these are three-dimensional structures, and so if you start messing with stuff starts moving around a lot. And so topoisomerase is going to help keep stuff kind of in line. Okay, so DNA helicase will unzip the DNA. Okay. All right, so this is going to allow the other enzymes to start to access those bases. So remember, our bases are on the inside of DNA. So like here, it's still connected right here. So we still have that double helix structure. Whereas back here, because this is where helicase is, we have separated the DNA. So again, it's like a zipper. Okay, in this diagram, the zipper is going from right to left. So as helicase moves this way, it starts to unzip the DNA molecule. Okay, and it says, no, you can't hang out. Adenine, you can't hang out with thymine anymore. Okay, so, excuse me. As we start to unwind the DNA, we're going to have our next enzyme, RNA Primase, come in. And RNA Primase, so a primer is going to be made, and that primer is going to be made from RNA. Okay, so this is an enzyme that makes what is called a primer. So like if you've ever painted a house or anything like that, if you want your paint to stick on and last a while, you want to put down what's called a primer. So it's something you put down first that allows the rest of the process to go a little bit better. And so this is named that for the exact same reason. We're going to make some RNA primers. Okay, so that's basically just like a little strip of RNA. So it's RNA, remember, so I'm not going to have any thymine. Now I've got uracil. So like instead of A having a T next to it when I make my primer, I'm going to have a U next to it. Okay. This is like my foundation. Now that I've made an RNA primase, so I've unzipped the DNA, and then my RNA primase has come in, and it's latched on right here. It's chosen this spot for a specific reason. Okay, so there's chemicals and chemical bond relationships that are going to dictate where it goes. It will only go to certain areas, and we'll talk about that later, but it's going to basically create a starting point. Okay, so it's going to create, in this case, a 4 nucleotide long primer. Okay, so that's what I've drawn here in the third diagram, or what I've circled in the third diagram. And that's like a starting point. This is where DNA polymerase, so now I've got another enzyme, it ends in ACE, so it's an enzyme. This is the one that polymerizes or chains together DNA. Okay. DNA polymerase is going to latch on to the RNA primer because DNA polymerase, it can only grab onto certain things. And one of the things it can grab onto is an RNA primer. So if I didn't do this, then nothing else down here could have happened. My DNA polymerase could not have attached, and thus we wouldn't be able to make DNA. Okay, so that's why we have to make this RNA primer. So even though it kind of makes everything mismatched, like now I've got these random uracils here instead of thymines, like this DNA looks funky, right? But it'll all get fixed. All right, so this is stage 2. We've unwound the DNA, we've made our RNA primer, and DNA polymerase has latched on to the RNA primer. And now we're going to have the synthesis stage. So synthesis is when you make something, right? So DNA is going to be made. And you guessed it, the enzyme that is responsible for making DNA is going to be DNA polymerase, right? And it's going to make a whole new strand of DNA. And it's going to follow the same rules you've seen already, A to T and C to G. Adenine likes to hang out with thymine, and cytosine likes to hang out with guanine. Okay? And so you'll notice here that the classic double helix structure gets a little more complicated when you start replicating, because here we have right here like the beginnings of a replication bubble. We're going to talk about what that is later in class. But then we have what looks like a DNA strand here, so a double helix here, and what will become another double helix here. So it's really like one double helix becoming 2. Right? And that should make sense because we're copying the DNA in its entirety. But it gets really complicated. Okay? And so DNA polymerase is going to add nucleotides. But here's the thing. DNA polymerase is a little limited. It can only add in the 5' to 3' direction. Okay? So remember how those DNA strands had directionality, right? We had a 5' that pointed towards the 3' end, and then the other strand did the same thing, except the arrow was flip-flopped. So it was like if one was pointing up, the other was pointing down, right? So that's great, but DNA polymerase can only go in the 5' to 3' direction, which basically means that as I unwind the DNA, which I'm doing in this image right here, stuff is going to get complicated, because if I'm going in one direction, everything works great. But if I'm going in the other direction, I'm going to have to go in pieces. So like if you look here, excuse me, this top diagram right here, this top double helix that's being formed, it looks pretty clean. It's like one cohesive piece. Like this whole strand has been created so far in one solid piece. And it is going to continue to do that. Right? It's going to keep going as the DNA helicase, which is not diagrammed here, but is right here, keeps unzipping. It's this guy right here, this DNA polymerase is just going to follow. Okay. Whereas down here, you can only go in the 5' to 3' direction. So this guy is going to start going towards the tail end, but then he's going to get stopped. And then this guy will have to pick up. And so basically one of these strands is going to be kind of lagging. It's going to be falling behind because you got to go in little pieces. And that's going to make a lot more sense as we diagram that together. I don't expect you to be an expert in it yet. Okay, it is really something that you need to see and manipulate yourself before you can really understand. But for now, just know that we have what's called a leading strand. So, excuse me. The leading strand is like the easier one. This is the one that DNA polymerase likes because it can just like pop on, right? And then it'll just follow the DNA helicase. So here I have the DNA helicase, and it's going in this direction. It's unzipping the genes, right? Unzipping the double helix. Okay, and then as it does that, DNA polymerase, which is right here, is just going to follow along. And because it's going from 5' to 3' of the strand it's making, then it can just follow and never get off those tracks, and everything's going to go great. But over here, I have a 5' to 3'. So this top strand points in this direction, right? Which basically means that when I make DNA, it's going to point in the opposite direction, right? Because it has to be anti-parallel. And so what's going to happen is the helicase is going to be unwinding the DNA, and then this guy is going to be trying to catch up the whole time. Because right here, the DNA polymerase can just follow behind the helicase. But in the top strand, so the top strand in this case is what we would call the lagging strand, Okay, if this is the leading strand being formed at the bottom, this top one is going to be the lagging strand. That one has to be done in pieces, and that's just because DNA polymerase kind of sucks. Like DNA polymerase is really limited. It can only go from 5' to 3' of the strand that it is synthesizing, of the strand it's making. So it's trying to make a strand that looks like that, but it's not going great because it has to keep going in pieces. Okay. And again, this is something that I expect you to get better at as we do it in class. But basically that leads to one of these being really, really nice. So the leading strand gets synthesized, or the leading strand gets copied really, really easily. Okay, so DNA polymerase just gets to kind of follow along with the helicase. Whereas down here on the lagging strand, we just have to go in bits and pieces. So it's discontinuous. It's super annoying. Okay, the cell does not like dealing with this, but you got to do what you got to do. You got to make your DNA. So as we go down and we keep creating these little fragments, those are called Okazaki fragments. It's named after a husband and wife Japanese scientist pair that discovered these fragments. As they start to form over and over and over again, eventually, if we want this to be a good DNA molecule, we're going to have to like connect these in some way, and that's going to be in step 3. Okay. But for now, we have this replication fork, okay, and it's like a fork in the road. That's why it's called that. Basically, helicase is going towards the left here, and that's unzipping the genes, and so that means that I can make this top leading strand really, really easily. I can just follow along, but this bottom strand I have to do in bits and pieces. As it moves, I can slowly add more and more, okay, and then I'm going to have to go in and remove all of these RNA primers, including the one up here on the leading strand, and then eventually like staple all of these little Okazaki fragments together. Okay, so this is also called elongation because we're elongating the DNA strand. We're synthesizing and elongating the DNA strand. So you should just be prepared to see either of those terms, either elongation or synthesis. And we still have this RNA primer. Every single time that DNA polymerase had to pop up, it needed an RNA primer in order to get on there. And so if it didn't have that, nothing would get done. Okay. All right, so DNA ligase is responsible for going in and basically stapling everything together. So I mentioned before that those Okazaki fragments need to be connected. They can't just be floating around all separate from one another. They need to get stapled together. And DNA ligase is going to be responsible for doing that. And I don't need you to understand too much about the overall process. Just know that it's basically a giant stapler. Now, you're going to see this term called semi-conservative. And basically, this whole DNA replication process is called semi-conservative because I had this original strand here, and then I'm going to do DNA replication, and that each of those those double helixes I'm going to make has 1/2 of the original DNA strand, right? And that's just because we split it down the middle and then we started making it. We have a new strand in each as well, but it basically just means that like this color right here, this brownish color, is conserved in each of those 2 daughter double helices. Okay, here's the overall process. You're going to see a lot of diagrams like this where like you picture the overall chromosome, you zoom into certain parts, and DNA polymerase is going to be here. It's usually shown like in a big old bulbous way because it's just going to keep going and going and going and making our DNA molecule. Helicase is usually shown like with a triangle because it's like unzipping the genes. It's like poking through, right? And then here we have another DNA polymerase molecule. So it's really the same molecule, except this one's for the lagging strand on the bottom. And so it's going to go left to right, left to right, left to right, over and over and over again, and forming those Okazaki fragments. Okay. I don't need you to worry about retroviruses. I have it in here just mainly as an interesting point that I'd like to talk about in class if there's time. But if you need a little bit of information, then you probably will. about DNA replication because it is so complex. Here are some supplemental resources that I would recommend, some good videos, and we are going to practice those a lot in class. So I don't need you to freak out about DNA replication yet, but just understand that it is involved and you're going to have to spend some time diagramming it. And we're going to do our foldable. So that takes us to the end of DNA structure and function. And next time we're going to be talking about gene expression.